Hello, everyone. Welcome to today's RNA Collab seminar. And for the first talk of this session, I am happy to introduce Professor Susanne Gerber. She is a professor of clinical genomics and bioinformatics at University Medical Center Mainz and head of the Computational Systems Genetics Group. Susanne's research combines computational biology and genomics to develop methods for analyzing complex sequencing and multi-omics data, with applications ranging from RNA biology to human disease. Today, she will talk about RNA modifications in the human genome. The floor is yours, Professor.
Thank you very much for the kind introduction. Share my screen. I hope you can see the screen.
Yes,
Yes.
we see your screen.
Okay, cool. Thank you very much. So good morning or good evening, depending on where you're currently listening. First of all, I would really like to thank the organizer for the invitation and also the opportunity to speak in line of the seminar today, and also for the very professional organization. I was really impressed here. So I would like to start my presentation with a little provocative question. So if I asked you what a normal human epitranscriptome on a normal healthy human looks like, so the landscape of chemical modifications on the RNA, could anyone give at least even a vague answer? I guess not, even though you're not able currently to raise the hand, I know, but actually not even I could really give an answer here, because currently there simply is no reference epitranscriptome available that we could use and just check out how a normal human looks like. And I consider this to be quite remarkable, because over the past 2 or 3 decades, scientists have built reference maps for nearly every molecular layer of the human cell. So we do have reference genomes via the Human Genome Project, with hundreds of thousands of genomes that can be aligned to and allow us to investigate whether there is a variant or a mutation somewhere. We do have transcriptomic atlases, we have epigenetic reference maps, protein atlases, and so on. And most of these even with tissue-specific or even down to single-cell resolution. However, one fundamental layer is currently still almost completely missing. As said, so we do not have a reference atlas of the human epitranscriptome at all. And on the other side, you are all RNA people. We know that the RNA modifications have an immense functional role and also co-regulate virtually every aspect of RNA biology. So they impact stability, degradation, interaction with other RNA binding proteins. We know that many of them are also altered in diseases, which makes them in principle extremely interesting targets for biomarker discovery. RNA therapeutics, it's also something that went through the news during the corona pandemic at the latest. And my lab, for example, is also involved in clinical diagnostics. And when a clinical geneticist identifies a genetic variant, the first question that we ask is very simple. Is this normal biological variation, or is this a disease? And we can answer these questions because we do have the human genome as a reference. But for the RNA modifications, we still cannot answer the equivalent questions. So we cannot even identify if a certain modification on a certain human being in a certain age is a variant or not, as we simply don't know what constitutes the normal epitranscriptome of a healthy person at a certain age. And so even less, we do not have the answer, how does this landscape changes during lifespan? So is there remodeling like the epigenetic remodeling that we know during lifespan or not? And this was exactly the motivation behind the work that I would like to present today. Before I introduce the cohort that we recruited in the recent 1.5 years, so maybe just a little bit background for those who are not already RNA modification enthusiasts. So as already said, RNA modifications are extremely diverse, and compared to DNA, where the epigenetic information is encoded primarily through DNA methylation and histone modifications, RNA has a much richer chemical vocabulary. So currently more than 170 different modifications are known across different RNA classes. These here are only some examples for the tRNA. As said, well, at 170 are known, and these are installed by dedicated writer enzymes. They can be recognized by specific readers, and in some cases, they can even be actively removed by erasers. And over the past decade, we have learned a tremendous amount about the molecular function of individual RNA modifications. However, most studies still have focused on either single modifications or on specific cell lines or individual disease settings. And the goal of my lab that I'm going to present now was rather than focusing on single modifications or a particular RNA to establish a clinically relevant sex and lifespan resolved atlas of the human epitranscriptome. And now I'm showing how we did our first steps towards changing this. Yes, so the following are, by the way, also unpublished data and work, and therefore I ask you to refrain from sharing this without prior consent before asking me. So how did we recruit the subjects? So the criteria were that they have to be strictly healthy. So we started with newborns, so cord blood from newborns that were scheduled for, or that came from scheduled C-section. So the baby is unstressed and where we could very easily, without any ethical problems, just take some blood from the cord. Then we recruited also about 55 children in the age of 0, so several months until 18 years old via the Children's Hospital. Here again, they were strictly healthy. They were only scheduled for minor routine surgeries such as dental braces, circumcisions, strabismus surgeries, removal of metal pins. So anything, but where they can be also considered as completely healthy and unstressed, so no accidents or so. And on the other side, they already had venous access for these surgeries, and also again without any ethical problems, we could also here take blood from little children without harming them additionally to the venous access they anyway had for the surgery. We recruited adults via community outreach, and also elderly via the gerontology that are, again, so elderly means that they are elderly but completely well and healthy, do not take any medications, and are very fit for their particular age. Altogether, we recruited currently 155 subjects, and they were strictly sex balanced. Maybe I just switch to the next slide to show a bit more details. To remove this here. Yeah, so what you can see here is the age and the sex distribution. These are all the males, these are all the females, each dot is one subject, and what you can see that we have very nicely recruited. So for example, in each and every age bin, we have recruited a high density of subjects. So the several newborns, then we have 1-year-old, 2-year-old, 3-year-old, 4, 5, 6, 7, 8, 9, 10, and so on. Same for the females, up to 18 years. We have seamlessly recruited during childhood, even sometimes oversampled, meaning 2 or 3 children of the same age. And we did this because we must assume that due to the developmental jumps in this age, that the most regulatory activity occurs during the early childhood. And also something that I will mention later, we also had family structures in the cohort, and whenever we had siblings or a mother-daughter relationship, we also recruited another non-related child to have not a strong bias inside. During the adulthood, we had maximum gaps of 3 years between the recruitment, so 21, 24, 27, and so on. And in old age, we again recruited very densely, because here again we can assume that from 70 years onwards, the differences in the epitranscriptome might become increasingly greater than in the normal adulthood. And yeah, as I already spoiled, we also had some family structures. We have identical twins. We have, for example, also one complete family tree, like a grandma, her daughter, her son, daughter with husband, and then the 3 grandchildren, which is also nice to investigate epitranscriptomic inheritance, and also one longitudinal sample of at least one selected subject, also others, but this was the longest ones, which was currently more than 14 time points during a year, with a gap of sometimes 1 to 2 months, and also 1 week where we sampled consecutive days, and also we tried several or 2 different tubes, namely PAX and EDTA, but this would go too much into detail. If you have any questions, I'm happy to answer here the difference that we observed. So then after taking the blood, a very strict protocol, we then extracted or isolated the PBMCs after exactly 2 hours from taking the blood. Then we extracted 3 aliquots for tRNA, mRNA, and ribosomal RNA, and sequenced all of this using nanopore sequencing. I don't want to explain the concept in detail, but I guess most of you already know that when nanopore sequencing, you can sequence the native RNA and can predict based on the raw current then the existence of certain modifications. For selected samples, we also did orthogonal validation using GLORY and ETAM, just to make sure that we also do not have too much false positives. This then ended up in a cohort that has still a focus on the European white phenotype, if I may say so, but we also had some further ancestry backgrounds included, but the idea was indeed for the start to focus as narrow as possible, to not have too many biases or too many latent factors in the cohort that might influence our observation. So as said, then we sequenced 3 different RNA types. And in the following, I will only focus on the mRNA, because this just be a nice overview here, or a short overview. And what this resulted in was first a very general overview about the modification landscape. I will make it a little bit bigger. So what you can see is that we predicted first what are the unique positions that we can predict per modification type. And unique means that one modification position was found in at least one of the 155 subjects. And here we ended up with 580,000 almost modifications for M6A, almost 200,000 for M5C. We predicted inosine, pseudouridine, and the 2' O-methylations. And then we were questioning, how are these distributed over the cohort? So is this completely random when they appear, or is there some kind of pattern? And for this, we started sorting them based on their appearance on at least one subject, on 2 subjects, and so on, or at least 100 subjects that carry this particular modification. And what can be seen here is that obviously those modifications that appear only once, like in one individual, or not appear, I would better say, that have been predicted by the RADO, so the modification prediction tool, have a very low stoichiometry, meaning there is just a little bit above 10%, and 10% is this line that I just added here. This was the threshold filter that we used to accept the modification or not. So what we conclude here, that most likely this unique modifications that appear only once or twice can be false positives. And here we need to be very careful of trying to interpret some biology into this. Interesting enough, however, those modifications that appear obviously very often, like in at least 100 of the subjects in the cohort, they do have a very high modification frequency, stoichiometry, modification level, as you want to name it. But this means that obviously those that are very common and very often found in the participants have also a very strong signal, a very high modification level. And then we were investigating further these common modifications that we call, so to say, a backbone of the cohort. So these are all that were really called in each and every subject from the newborn up to the elderly. And these are almost 30,000 modifications on isoform level. And we also then investigated whether we could confirm these modifications via an orthogonal approach. In this case, it was glory sequencing, and the overlap is actually quite high. So almost 20,000 modifications could also be confirmed on isoform level. But then we mapped them on the genomic coordinates, meaning kind of concatenating the isoforms, which ends up with 44,000 modifications that are robustly or conserved across each and every subject. And here we could even confirm 36,000 or more than 36,000 modifications. This, however, does not mean that there's now a discrepancy, that they kind of exclude each other, or that there's a kind of a conflict. This could also just be based on the protocol differences, or also maybe in one of the two technologies there have not maybe even been enough coverage to predict the modification. So we did not dive too much into this, but this was already quite impressive that the overlap was here quite nice. And then we were asking whether these, again, only those conserved that appear in each and every subject, how they are distributed over the genomic location. So still we are on RNA level, right? But if you kind of project the RNA to its genomic coordinate, you can see that there are really some dense, so to say, regions where more than 100 M6A sites per megabase appear, compared to other regions that are really quite sparse. And obviously there's no transcript that could be that carried a modification that could be predicted. However, this is only for the M6A level. So summing up these observations, from what we could observe here was that there's obviously in the live constant conserved epitranscriptomic backbone, and that those modifications are also very high stoichiometry, meaning sporadic sites have, as I show you, predominantly low stoichiometry and can be also quite high certainty for being false positives. And also RNA modifications somehow cluster into reproducible hotspots or some kind of deserts that reveal a somehow organized epitranscriptomic landscape. So based on these findings, we then asked ourselves whether we could predict something like age, or whether we can see an age drift or an age remodeling. And for this, we started using just plain PCA on this cohort data. Again, each dot is a subject. And the PCA here shows, when coloring it in the live or in age, that obviously the newborns do somehow cluster together. These are also newborns, but otherwise, well, yes, you could say these are the children, but there's not really a clear pattern inside. Same if we clustered for age groups, meaning like children in a sense of 1 to 20 years, also children, adolescent, young adults, adults, and then 60 plus. But also here it more looks like a bunch of smarties, but not really that you could see a nice pattern in the global M6A level. Then we also were asking whether we can predict health status or see anything if we investigate the health status of these subjects. And that's why we also added samples of a cancer patient or from a chronic fatigue syndrome patient, but they, on the global level, you really do not see any kind of strong differences. However, I mean, we cannot conclude that this suggests that aging and sex do not globally remodel the epitranscriptome, but instead we were hypothesizing that the effects are likely localized to specific subsets of modification sites and therefore are rather site-specific. And then we were going deeper into this data set. I'm trying to hurry up a little bit based on the time. And what we could indeed find is that there are several of these spots of these site-specific locations where you can definitely see an increase in the stoichiometry in age. And also, if you do the similar way that you would do the volcano plots for gene expression, but here for modification level, you could also see that, for example, for very narrow age groups, so in those that are quite close together, like the age group 18 to 25 years old versus 26 to 39 years, there's only minimal differences that you could observe. However, if you do the same analysis and compare modification level from adults versus young children, you can see that there's really a strong signal and lots of up or down modified positions. And these findings suggest that obviously, indeed, the human epitranscriptome seems to undergo a pronounced remodeling during the early development and also during the late life. And this can be separated by, or this can be maybe explained by some kind of relative epitranscriptomic stability. And next, we were then asking whether we could identify genes with multiple sites that are obviously highly age-related, and whether we could use this as some kind of age predictor, so similar to the epigenetic clock from Steve Horvath that kind of I think everybody knows. And indeed, there were several genes, such as, for example, this LCKs, the live lymphocyte-specific protein, tyrosine kinase, that has really 4 different locations, actually is even 8 on isoform level, that are all related with age and all show a strong age drift. And we selected approximately 200 of these age-related genes. And what you can see here is not a basic PCA, but a so-called supervised PCA, where we're asking whether or not we can see an age drift. And what you can see here is that obviously, if you select particular age-related modification sites on about approximately 200 genes, so if you just narrow down your search space, then you can have really a very nice predictor. I don't know whether you can see this very well, but the lower, so this is kind of the true age from 0 to 80 years old. So the lower these the dots are, the younger the child is indeed, and the color shows the predicted color of the model. And this, by the way, just a statistical model, so a linear statistical model, so no AI, no 250,000 dimensions. It's really a statistical linear model that shows that you can predict aging based on the epitranscriptome. And the stars, by the way, are those that have not even been seen by the first PCA, but they have just been predicted after the fitting. We also then used some machine learning based approaches, and this was some entropy based approach, random forward gradient boosting, multilinear regression, and TAP-PFN. And what you can also see here nicely, even though there is definitely some plus minus divergence from the predicted, so the gray ones is the true age and the red ones is the predicted model. But here I also have to say that the model was only trained on a very small subset, so I think 40 subjects only at the beginning. So the more subjects you include, of course, the better it is, but we had to hold back several of the subjects to show how good the model performs on untrained data. And this is, I think, almost the last slide I do have. So if you now use this machine learning model, then you can, this sort of surface plot here can illustrate that this decision surface that can be learned by a machine learning model can really show that there's a drift in the epitranscriptomic landscape. So what you can see here in principle is that the shift from very young age, so everything on this plateau here are the very small children. Here and this plateau are adults between 30 and 50 years old, and here are the elderly. That there's really a transition from here to there. That and these are the 200 genes that we use to train this model, and obviously these are the modification sites that decrease during this aging process from child to the adult, and these all are those where the modification level increases from childhood to adulthood. And these are the same. So here they are nicely sorted. That's why the curve is so plain. Here it's the same modification. What you can see here, that those modifications that have been kind of strongly decreasing in this process from child to adult are those that now start increasing. Some of them still decrease, but interestingly enough, those that were at the first transition rather stable, they are those that are now going either become extremely high or extremely down modified, while others are still either upregulated or even going down. So what I would like to summarize here is, first, I mean, we are just starting to understand and to scratch at the surface of this data. So as said, it's not published yet, and it's also not that we are done with our analysis, but I think we can already conclude that the behavior or the aging is here really associated with a uniform gain or loss of RNA modifications, and the so similar to the epigenetic clock, there are really some trajectories that increase or decrease with age, and it's not just some on and off of certain genes that we can observe here. And maybe summarizing what I said before, so what we can conclude from our observations is that the epitranscriptome in the healthy human during lifetime really builds upon a stable backbone of highly conserved RNA modification. Obviously, aging leaves reproducible and also site-specific signatures across the epitranscriptome, so similar to the epigenetic remodeling. And these signatures can even be used to build an epitranscriptomic clock and predict age just based on a certain modification or an epitranscriptomic sample. And what we see is that these kind of reference atlas that are currently emerging can have really now the potential to open the door to clinically interpretable RNA modification biomarkers. Thank you very much for your attention. The highlighted students are those that are really the major driver of this work, and I would like to thank the funding agencies and also you for your attention, and I'm happy to answer questions if there are any.
Thank you very much, Dr. Susanne. Yes, so we have time for a few short questions, and I will read them to you now. So, starting with the question from Kong, can the differences between age groups in your analysis be simply explained by differences in gene or mRNA expression levels? Thanks.
How we define the age groups? Sorry, I did not really understand the question. So if the question was how we defined the age groups, well, we used the schematics that is normal also in the clinical environment, where you have this newborn, then you have the children, so the small children up to 5 years, then you have normally the gap from 5 years to 12, then you have the adolescent, then you have the young adults, the adults, and the elderly. What we, however, also observed that because we indeed also had the gene expression profiles, I did not talk at all about this, but yes, we had the complete gene expression profiles for each subject, and here you could also see changes in the gene expression, but particularly between the newborns and the children's. So therefore, I would say the boundaries were not chosen by us based on the data that we saw. It was more that we had the classical way of defining the age groups, and we only actually used the age groups only once in this one figure way. I wanted to show that on the global level, you don't even really see the age groups. So what we recruited was children from each and every age for each sex, and the classification for age groups is maybe something that will become handy and important later if you use this for clinical diagnostics, but then you can define your own age group. So we will also provide the data via a browsable tool, so an online tool where you can then also classify your groups that you're interested in. So if you, for example, have now your patient, a 5-year-old girl, then you can select also from the web browser all children that are aged and sex matched in a certain range. I hope this answers the question. Mm-hmm.
So another question from Chris now. How tissue specific are your modifications? Your RNA samples were all based on PBMCs. Have you tried to compare the modification signature from a given age group to other tissues from age-matched samples, if available?
No, we did not compare to different tissues. What we did, however, was a PBMC deconvolution. So you can, based on single cell data, there are several tools available. And also we had for several samples, we had also DNA methylation data. This allows you to do a deconvolution and see the proportions of the cells that are in your PBMC bulk, so to say. And this is something that we can also compare against each other, but the tissue itself was not available. As said, there were only healthy children, and we were only, also due to ethical standards, we were only allowed to take some blood from the kids.
Okay, so time for just one more quick one. Have you seen significant shifts in tRNA or RNA modifications with age so far?
In the rRNAs, the tRNAs we did not touch so far because, we had at the beginning a little bit problems with the I wouldn't say problems, but we are a little bit careful with making predictions on tRNA, as this is still quite noisy for a nanopore sequencing. Here we first really wanted to also validate and make sure that we can trust the predicted modifications. On the ribosomal RNA, yes, the differences are even larger, but this is something that we are also currently just digging in. If anyone is interested in this study and is also maybe an RNA expert more than us in the ribosomal RNA, you can also just contact us, and then I'm also happy to discuss further details here.
Okay. I'm sorry. So for the sake of time, maybe we'll call it quits here. And thank you very much, Susanne, for the incredible talk. Now I'll pass the spotlight to Joanna and Kinga.
So good morning. Today I have a great pleasure to introduce our next speaker, Dr. Kinga Kamieniarz-Gdula. So after undergraduate studies at Mickiewicz University, Kinga spent 13 years abroad working at Max Planck Institutes in Freiburg, as well as the Oxford University. Kinga returned to Poznań in 2019 to establish her own research group at Center for Advanced Technologies. Today we will hear a story about pre-mRNA pre-prime end processing and transcription termination. Kinga, the floor is yours.
Thank you for the nice introduction. Can you see my slides?
Yes.
You can see my slide. Okay, great. Sorry, just didn't put on the pointer. Yes. So as Joanna mentioned, I'm at Mickiewicz University in Poznań, and I would like. But can you see it properly? Because I have something in front. But maybe do you see my slide properly, or do you see a black bar on the top?
I can see them properly.
Okay, so this is just on my screen. That's fine then. So I would actually like to start by acknowledging the people that that this work is due to. I will show some data that has primarily been the work of Agata Stempien and Deepshika Pulimamidi, although actually most of other lab members have helped, especially in the last couple of months when we've been revising our manuscript relating to this data. I'm thankful to our collaborators and our founding bodies for support. So we are really interested in gene architecture, and the beginning of the gene is really neat and clean, also in terms of its definition, because we basically define the beginning of the gene by the position in which the enzyme RNA polymerase II, which transcribed all human protein coding genes as well as many non-coding genes. And so this first nucleotide that is copied into RNA is protected by this cap structure, and it actually is then maintained in the mature mRNA. So this is a very nice and clean definition. Gene ends are much more messy. They are much more messy because there are two processes that happen there in close proximity, both spatial and time-wise, but still they are not one. So the first one is the cleavage of the 3' end of the pre-mRNA and transcription termination. And while the beginning of the gene is annotated due to transcription, but actually the end annotation of the end is due to RNA processing. We define this as the position in which the nascent RNA is cleaved by the cleavage and polyadenylation complex. So to this position, then the poly A tail is added, thus creating the mature messenger RNA. But RNA polymerase II continues transcribing, and in case of the human genome, this happens for a median of 1,000 base pairs further. For some genes, can extend even much further. So in case of what is also important is that both of those processes that occur at gene ends, they are really relevant physiologically. So in terms of the pre-mRNA 3' cleavage, I mean, this very directly efficiency of this cleavage will very directly influence the efficiency of the output of the amount of mRNA that we will get. But besides the efficiencies, what is also important is that most human protein coding genes, they don't have one cleavage site, they have multiple potential alternative cleavage and polyadenylation sites. And this is actually relevant physiologically. A classic example is the heavy chain of immunoglobulin IgM, which can be either in a soluble, have either a soluble or membrane-bound form, depending on which a cleavage site is used. But unfortunately, also using different cleavage sites can lead to disease. For example, certain genes that have proto-oncogene characteristics, they might actually become more highly expressed and become an active oncogene if a different, quite usually proximal pollination site is used. So cleavage is definitely important, but termination itself as well. So usually when we think of transcription, we might be thinking of the polymerase binding, going from the beginning to the end, and that's it. But actually, many protein coding genes can be prematurely terminated. This is sometimes also referred to as transcription attenuation, and it is a process which allows to also regulate in a very precise way the amount of RNA that is produced from a gene. And this is something that was known for very many years from work from multiple wonderful yeast labs. And as usual, metazoans were a bit realized this a bit later, but we now also realize that in fact this does also happen, that premature transcription termination can also be a regulatory mechanism in mammalian cells, and there are several factors that can trigger this. One of them is, for example, a classic termination factor called PCF11, but also a very strong premature termination activity of the integrator complex has been demonstrated. But besides premature termination, termination is important also for other things. I mean, we need to recycle the polymerase for subsequent genes of transcription, but also gene punctuation is important. If transcription doesn't stop at the right place, then we can get interference with a neighboring gene. And also another thing worth kind of remembering is that most of the genome can be transcribed at some point. So we have a huge noise of a lot of non-coding transcription. So this also has to be reduced where it's not functionally needed at high levels. And connected with this, we start realizing that that transcription termination can be connected with diseases. This is a summary that was inspired by recent work by Carla Neugebauer's lab that studied very early effects of cancer treatment with imatinib. If you haven't seen it, I really encourage everyone to read. I think it's a really important paper showing that already after 1 hour of treatment with this drug, the very first molecular event that happens is actually delayed termination, and changes in gene expression, other changes happen later on. Okay, so what we study, I believe, is quite important. Now, I myself have been until quite recently, and I think many people might have the simplified picture, is that if there are any changes happening in 3'RNA cleavage or termination, that they should be going in the same direction, meaning that if we have more downstream cleavage, the termination should be also more downstream, and if we have proximal choice of cleavage site, that would be rather connected with earlier termination. So basically, what I will show you right now is that it turns out that, at least in the model that we have been looking, but we looked also in some other models that I won't have actually time to discuss today, it seems that this isn't exactly the case. So as I mentioned, so this is work primarily of a postdoc in the lab, Agata Stępień, primarily wet lab scientist, and computational PhD student Deepshika Pulimamidi. And the two main characters here are two proteins that are really key for transcription termination, but they're also involved in 3' processing. The first protein, CPSF73, is the endonuclease, which is the protein which cleaves the pre-mRNA at the 3' end, but it doesn't do it by itself. It needs this huge complex help with specificity and not also just to cleave all the RNAs that are around in the nucleus. So this is like a key endonuclease, but it's also really, this cleavage event is really important for termination. Without it, we see huge delays in termination. And the second factor is PCF11. This is a factor which is substoichiometric to the rest of this complex. So it appears it might have some regulatory function, and besides, so it enhances efficiency in cleavage. In fact, in vitro, we cannot get any cleavage without it so far. And but on the other hand, it's the only protein that has been shown to have the capacity to dismantle an elongating polymerase. So by some nice experiments done in Gilmore lab. So these are our two main heroes. Now, I said I'll be talking about the cancer. So what is the relevance in cancer? Well, nice studies from Sven Dankwart lab and also from Mike Vane's lab have shown that in the case of neuroblastoma and here pancreatic cancer, high levels of those factors are actually predictive of very poor outcome for the patients. Another piece of evidence suggesting the clinical relevance here was the cancer level was the fact that a compound which was considered a promising drug target because it cures cancer, at least in mice in some cell culture model. When a consortium tried to find what the real molecular target was, it turned out that CP70 is actually the primary molecular molecular target. So we see that there could be some therapeutic opportunity here. Something that is not really clear is whether these oncogenic properties of those factors are, because they act both in cleavage and termination, are both those processes involved in this or not. So this was entirely unknown. And in this study, we decided to focus on colorectal cancer as a model. So colorectal cancer is a cancer which is third most common in the world and also second most lethal. So it's a big problem. And we found it very interesting that when looking in tissue samples from patients, it was clear that the protein levels of all factors that we looked at, and both in 3' processing and termination, were upregulated. So the model we decided to use consists of 4 cell lines that we did most of the experiments with. The one CT cell which comes from the healthy colorectal epithelium, then 2 cell lines derived from the primary tumor, and then a metastatic cell line which is derived from the same patient as W418, but after metastasis. And actually those 2 paired cell lines were our main workhorse, but we also looked at those other ones. And what was really nice is that we could see that those cell lines recapitulated the levels, the increased levels of processing termination factors in cancer compared to normal tissue, which was also seen in biopsies from patients. So what we did not know was whether in the context of CRCC, of colorectal cancer, CRC, those factors acted as oncogenes, because this wasn't tested. So we tested this with colony formation assays. And basically what we could see is that when performing knockdowns, we could see a very strong effect on the cell line coming from primary tumor, as only a slight one, only with PCF11 with using the cell line from metastasis. And consistently with this, when using the inhibitor, JT607, we could also see that while both cell lines responded, there was clearly a much stronger response of the cell line from primary cancer. we confirmed this now also with some other examples of primary metastatic colorectal cancer. So what we basically saw is that that primary tumor cells have much higher sensitivity to both CPA downregulation and CPA inhibition. So to conclude, we concluded that the levels of termination factors matter to colorectal cancer cells, but also that to our surprise, the primary tumor-derived cells were particularly sensitive. And we were wondering why this could be. And because for the sake of time, I will run a bit fast about two hypotheses that we had and tested, but turned out not to be the case. So one hypothesis that we were really betting, we were really confident this was what would be happening, because this was also something that was suggested in literature, although not tested. We thought that what would happen is that when we inhibit or downregulate termination factors, we will have increase in transcription replication collisions. And this, we had an idea how this could contribute to the sensitivity of the cells. And so we did really a lot of experiments in collaboration with the expert in the field, so Stefan Hamperl and Maxime Lalonde. We used the PLA assay to test the transcription replication collisions using antibodies against PCNA and against terminating form of POL2, but we did it also with just an elongating serine 2-phosphorylated form of POL2. And so I'm just showing a few experiments, but in general, what we were expecting was seeing a significant increase in collisions upon knockdown or inhibition of those factors, but neither using knockdown nor inhibition nor degrons, we could not see anything significant. And if any tendency we could see, then it was even in the opposite direction. What was quite interesting is we saw for the first time we could see what happens during, let's say, in a model of cancer progression to transcription replication conflicts, and it seems that they are downregulating during cancer progression, at least in the case of this particular patient between the primary and the metastatic cancer. But this beautiful hypothesis was not true. Another hypothesis we tested was also based in some suggested literature that suggested that increase in CPA factor levels would lead to shorter mRNA isoforms, and that shorter mRNA isoforms would make cells more sensitive to CPA73 inhibition. Again, this turned absolutely not to be the case. So in our case, we did 3' mRNA sequence, so we determined where cleavage and pollination mainly occurred. So as we see, the primary cells that are more sensitive, if there is any, for half of gene, we didn't see a shift, but if there was a shift, it tended to be actually lengthening, not shortening. So this was one thing that didn't fit with this hypothesis. And another thing that doesn't fit either. So we really did a lot of analysis. We really tried very hard to see any association of alternative polyadenylation with this with the cancerogenic process here, but we couldn't see it. So, for example, if you look at changes that occur on oncogenes versus tumor suppressor genes, they're exactly the same as for all other active protein coding genes. So it didn't seem like somehow this is really functionally important in this context, or that this would make cells particularly sensitive. So we turned to look in more detail at transcription termination, and the method that we particularly like in the lab is a method developed by my colleague Takanoima when we were both postdoc in Nick Ploutford lab. So this is a method called mNETseq, and particularly if we use this method with an antibody which is directed against threonine 4 in the heptad repeats of the CTG of POL2. This very beautifully highlights where termination occurs, or terminal pausing occurs, both at the end of gene but also in the gene body. And you can see, if you compare to other nascent transcriptomic methods, there is some advantage when looking at gene ends, because it's easier to quantify an increase in signal than kind of really find where exactly which decrease to look at. But it's particularly helpful also for premature termination happening in the gene bodies. So we use analytics both with total polymerase and with this termination-specific mark. And we could see in both cases, but with better kind of view and resolution with the we could see something really surprising again, which was that the cells from primary tumor, they showed accelerated termination compared to the control cells, which are black. And then another super surprising was that actually the metastatic cells, they showed like a partial reversal in the direction of what is happening in the cells from healthy epithelium. And this can be seen here also so this is an averaged meta gene, and you can see this on the example gene that there is this earlier termination that is partially reversing. But if you look here in the gene body, what you can also see that there's also an increase of premature termination in cancer cells, particularly in the cell lines that are derived from primary tumors, but also the metastatic ones compared to 1CG. Again, this is some single gene examples which show this increase. But the striking thing where I said that it was surprising to see termination happen earlier in cells from primary tumor was the fact that we saw cleavage occurring later. We saw delay. We saw lengthening of mRNA, and here we see here we see earlier termination. So we thought maybe we have to really look into the different functional categories. In different categories of depending where cleavage is occurring, whether it's happening moving proximally or distally, but actually no, this is not the case. It doesn't matter if we look at genes where an earlier pollination site have been cleaved or later. We always see that termination happens earlier than in cells from healthy epithelium, and there's a partial reversal in metastatic, and this is the case for all categories we saw. And this is also an example that shows that shows on a single gene. So basically, we don't see like a unidirectional change. We see that there's something happening in both directions here, or actually we see changes also in opposite directions here. And in particular, so if we have like cells from healthy correct epithelium. We have cleavage and termination occurring at a certain distance to each other. What we observed in cells from primary tumor is that the same cleavage sites might be used, or more distal one, generating a longer mRNA, but termination is accelerated. While in the case of the cells from metastasis, we see an opposite trend both for cleavage and for termination, the partial reversal to the normal phenotypes. So this prompted us to look at two things as an additional experiment. So one was we wondered, does it really matter, let's say, to the expression of the gene, this distance between those two processes? So first we quantified what we could see already in the averaged plots, meta plots, the distance between cleavage and termination. So we could see that there's a very strong decrease in this distance in cells from primary tumors, a partial reversal here. And what is also really striking is that this is this distance is much shorter for oncogenes. So in the end, between the average distance that we see in cells from healthy epithelium and in oncogenes in cancer cells, there's like twofold closer distance. And And what we also saw was a mild anticorrelation, mild because the R coefficient is very small, but this is still a significant anticorrelation between gene expression and cleavage termination distance. When I say mild, it is still much stronger than the influence of alternative pollination on gene expression in the same system. And what this shows is that the shorter the distance, the higher the gene expression. So if we had a shortening of the distance from the normal epithelium to the tumor cells, then at the same time we tend to have increased gene expression. So this is comparing two cell lines with each other, but also if we look just within one cell line, we can see a similar thing. So so if we look at genes that where a cleavage and termination happen closer to each other, they tend to have higher expression than genes where it's further and further apart. So of course, these are just correlations. We wanted at least to challenge the system a little bit. For this, we knocked down PCF11. The effect of this was that the distance between cleavage and termination increased. So the effect on termination was stronger. This distance increased, and in fact, the genes we could see also on both on individual genes and also looking at genes in bulk. That if we have if we see an anticorrelation of the distance between cleavage and termination and the expression of the gene. And I have to say that expression here we measured by 3'RNA-seq done on nuclear RNA to look at what is happening in the nucleus, not at let's say effect of stability in the cytoplasm and things like this. Okay, so I already summarized this, but another thing, so we now saw that this decreased distance seems to correlate with increased expression. Another thing that we thought of is it possible that on one hand it allows maybe the primary tumor to upregulate oncogenes, but maybe this fact that they occur so close to each other that somehow it might make the cells more sensitive to GTE. So to test this hypothesis, we decided to look at a very different system, pancreatic cells that were studied in the Fengin lab, which showed that the cell line MAIA-PACA2 was relatively resistant to GTE, whereas PANC1 was very sensitive. So we also performed NGS, both 3'RNA-seq and threonine-4-MNET-seq on those cell lines. And what we found that fitting with our hypothesis that the sensitive cells have a shorter cleavage termination distance compared to the cells that have the pancreatic cancer cell line that are more resistant, and this is like an example gene that shows this too. So of course this doesn't prove this, but I think it's really encouraging to do much broader and systematic study in this respect. Okay, so I wanted to mention some other tools in the lab. So basically we mainly use nascent transcriptomic, but we also do a single molecular RNA fish and some microscopy screening with this, and reconstitute pre-mRNA 3' cleavage in vitro, which is done by Martyna Pląskowska and Joanna Kwiatkowska. And this is my last slide. So what I'd like you to take home from my presentation is, first of all, that gene sense, while they're messy, they are important and worth looking at. And secondly, while pre-mRNA 3' cleavage and transcription termination are coupled, for example, as I mentioned, that they share a set of proteins that is important for both processes, and there's much more experimental evidence for the coupling. But it turns out that the coupling does not work in a way that that we've been thinking in that the two processes would, when they change, that they would change in the same direction. It rather seems that basically all directions are possible. So with this, I'd like to thank you for your attention. I'm happy to take any questions.
Thank you, Kinga, for your presentation. We have one question. Thank you for a great talk. Is it possible that there is increased premature cleavage and polyadenylation in your CRC cells, but those short transcripts are degraded, leading you to mostly see the distally shifted apophytes in your RNA-seq data? How did you control for this in your analysis?
So we do see. So in general, we do see more premature termination. That is both looking at cleavage and polyadenylation and transcription termination. So this is something that we kind of schematically show here, but this is something that we can see also on particular on particular genes. I mean, how can I or here are examples that we can clearly see that there's more premature termination happening in cells from cancer than in the cells from the healthy colorectal epithelium. So this is the case, but let's say there are two independent things. So and what is actually one of the things that would be contradicting this, that we still see more. I mean, we still see quite a lot of. We see the premature termination dependent if the termination moves at the gene end, moves a bit more proximal, more distal. I don't know whether this help or I should give some more follow-up. So in general, the manuscript is available as a preprint. Hopefully it will be also accepted as a publication, so all the kind of details of how the analysis was done. But as far as we could see, this of course we cannot rule out that there is some termination that we cannot detect, but as far as we could detect with our methods, this did not seem to be influencing our results.
Okay, and we have one more question, the last one. Why does the termination site affect gene expression? Do these fragments after the cleavage site have functions in the cell?
I don't know of functions of the downstream. So the downstream fragments are in general very unstable, very quickly degraded. And I cannot think, there might be something, but nothing comes to my mind at the moment of a functional that would be known. What is the kind of for a particular for any particular process? So why this we really don't know. So is the let's say the honest and biggest answer. So one of the hypotheses we have is that maybe if those two processes occur close to each other, that the cleavage would be more efficient, and for this reason there will be the gene expression would be going up. But we don't have experimental evidence that would kind of so far that would really prove this.
Okay, thank you, Kinga, for your presentation, and thank you all for joining us today, especially to our speakers for their excellent presentations and engaging discussions. Before we close, I would like to remind you that our next seminar will take place on August 12th at 10 a.m. Eastern Time, hosted by Zhejiang University Center for RNA Medicine and the RNA Institute of the State University of New York, Albany. Hope you will be able to join us again. Thank you very much for your attention.